Great. So, hello everyone. Welcome to our Kaigangan Biodiversity Research Webinar Series, and I am Elaine Villanueva, your host for today. And this webinar aims to raise awareness on the conservation and sustainable use of Kaigangan biodiversity through a series of online talks from our very own experts. So this webinar is made possible by CONserve-KAIGANGAN, a research program of the University of the Philippines, Los Baños, and Samar State University, funded by DOST under the Grants in Aid program. And so, in case you missed our previous webinars, okay, yeah. In case you missed them, you can you can watch them on our YouTube. We have our YouTube channel, and this is the link to our playlist. So you can go to bit.ly/kaigangan playlist. That's bit.ly/kaigangan playlist. And again, we would like to say hello to our audience, and please feel free to introduce yourselves using our chat box and to our comments on Facebook Live. So just like to remind you before we begin our webinar, so we have our guidelines here. So first, please keep your microphones off. Second is you can send your questions for our speaker via chat using the chat box from Zoom or Facebook comments. And lastly, we would like to request everyone to fill out our evaluation forms via Google Forms. So the link for that will be released later, and certificates will be sent to the email indicated in the forms. Okay. So to begin, let's start by introducing our speaker for today. So he is an assistant professor at the Microbiology Division of the Institute of Biological Sciences, UPLB, and he finished his master's degree in microbiology, minor in molecular biology and biotechnology. And he is also a specialist microbiologist and a proud member of Phi Sigma Honor Society and the Philippine Society for Microbiology. He has published a paper entitled Characterization of Plant Growth Promoting Bioisotrophs from Salt-Affected Areas in the Philippines, and co-authored another entitled Sequence Analysis of New Variants of Porcine Epidemic Diarrhea Virus in Luzon, Philippines in 2017. Let us all welcome and show our virtual clapping reactions to our speaker for today, Assistant Professor John Daniel O.
Thank you, Elaine, for that kind introduction. So let me just share my slide. Can you see my slide, Elaine?
Yes, sir.
All right. So good morning, everyone. As was mentioned by Elaine, I am John Daniel P. Ong, a faculty of the Microbiology Division, Institute of Biological Sciences, UPLB, and a member of the DOST-funded program, Assessment and Conservation of Forests over Limestone Ecosystem Biodiversity in Selected Municipalities of Samar Island, Philippines, also known as CONserve-KAIGANGAN. Now, before we talk about the topic this morning, let me first introduce our program leader and the members of the Project 3 team. Leading the program is Dr. Inocencio E. Buot Jr. Dr. Marilyn P. Balolong and Professor Edmerson B. Geronimo are our project staff. Ms. Yula Francia Embosito is our Science Research Specialist. Ms. Florabel D. Kerubin is our University Research Associate 2 and MSMBB student from UPLB working with the project. Christian Gabriel D. M. Villarama is our University Research Associate 1. Another student working with the project is Ms. Crystal Christiame P. Gonzalez, an MS Microbiology student from UPLB. And actually, we have 4 BS Biology students from UP Manila working with the project, and they are being mentored by Dr. Marilyn P. Balolo. We have Zachary B. Lara, Clariel Dane Q. Leonardo, Maria Julia Catherine H. David, and Micaela Maria S. Mahusay. Now, in this morning, we will be talking about the first report on the profiling of soil bacterial and fungal communities in karst forest of Samar using next generation sequencing. Now, just a reminder, again, this is the first study, and the um, the results are not yet published. So I request the participants to refrain from um, making screenshots of the results. So other um, slides, okay lang. But for the results, please refrain from taking a screenshot. Thank you. Now, forest over limestone, or more commonly known as karst ecosystems, is a type of land formation within tropical rainforest regions along limestone hill areas. Now, they are one of the most ecologically fragile ecosystems in the world. These landscapes are actually threatened by anthropogenic activities and natural processes. Karst ecosystems are typically thin, coarse, highly erosive, and degenerative. Now, in this image right here, you can see these are images that we took from when we were doing reconnaissance survey of the area, and we actually observe erosion in certain areas of the karst forest, as you can see in the figure at the right. Now, nearly 25% of the world's population lives in karst areas, and the Philippines has been recorded to have a vast area of karst or forest over limestone landscapes, but a large portion of it remains unstudied and unprotected. Now, it covers about 30,000 square kilometers, or 10% of the total land area of the Philippines. And karst soils have excessive amounts of exchangeable calcium that limit the growth of certain vegetation. Now, the adaptive capability of plants to grow in such an environment partially or completely comes from symbiotic or associative relationship with microorganisms. Well, soil microorganisms help in recovering and maintaining the health of ecosystems, particularly karst ecosystems. Now, soil microbial communities also play an important role in the release and retention of soil nutrients and soil fertility. These microorganisms also play an essential role in shaping the biodiversity and the functioning of terrestrial ecosystems by driving biogeochemical processes and mediating nutrient turnover. Now, other uses of soil microbes are shown. So you have production of phytohormones. You also have nutrient cycling, detoxification of contaminated soil, biocontrol of soil-borne phytopathogens, and organic matter decomposition. However, soil microbial processes and microbial resource limitation of a karst ecosystem remain poorly understood. Now, in this study, as I've mentioned, we report for the first time the bacterial and fungal metagenomic snapshot of forest over limestone in the Philippines using next generation sequencing and metagenomic analysis. Now, this gives us an insight or insights into the abundance, diversity, and metabolic capabilities of microbial communities in forest over limestone ecosystems in Samar, Philippines, and their possible roles in these karst ecosystems in terms of ecosystem health. Now, before we go into the results of the study, let me just discuss a little bit about NGS, or next generation sequencing, and the metagenomic workflow. Now, in principle, the concepts behind Sanger sequencing and NGS, or next generation sequencing technologies, are actually similar. In both NGS and Sanger sequencing, which is also known as dideoxy or capillary electrophoresis sequencing, the DNA polymerase adds fluorescent nucleotides one by one into a growing template or DNA template strand. Now, each incorporated nucleotide is then identified by its fluorescent tag. Now, the critical difference between the two is that Sanger sequencing only sequences a single DNA fragment at a time. Now, NGS, on the other hand, is massively parallel, meaning it sequences millions of fragments simultaneously per run. Now, this high throughput process translates into sequencing hundreds to thousands of genes at one single time. NGS also offers greater discovery power to detect novel or rare variants with deep sequencing. Now, here's just a table showing you the benefits and challenges in Sanger and NGS. So for Sanger sequencing, it is fast and cost-effective when sequencing low number of targets. Now, however, it has low sensitivity, low discovery power, and not as cost-effective when dealing with high number of targets. Whereas NGS has higher sequencing depth, which enables higher sensitivity, and it also has higher discovery power, meaning it has a higher chance of identifying novel variants, then higher mutation resolution, more data is produced, and there is high sample throughput. But the challenges when it comes to NGS would be less cost-effective and time-consuming when dealing with low number of targets. Now, here is an example of a picture of a metagenomic workflow or a metagenomic pipeline. Now, as you can see, it's a little bit complex, but let me just simplify things for you by showing you this image that I obtained from two of our UP Manila students. Okay, so mas pinasimple itong image na to, mas maintindihan natin. So we have here the metagenomic pipeline. An analysis pipeline actually, or metagenomic pipeline, refers to a particular program or script that combines several or even dozens of software programs to complete a complex analysis task. Now, before doing metagenomic analysis, usually what you do, or what you should do actually, is to establish your metagenomic pipeline. And this specific method, or this specific task, is usually overlooked in many different experiments. So ang nangyayari na lang minsan would be they use random softwares to perform analysis dun sa kanilang mga raw sequences without really understanding or without really developing or establishing the proper metagenomic pipeline for their specific sample. Now, how would you do this, you might ask? So what you could do is to review many journal articles which are similar to the study that you are doing, and you can put together several pipelines. You can combine them or even use the pipelines that were previously used by other authors or other scientists. Now, once you are done with sequencing, you obtain raw reads. Okay? Now, high throughput sequencing reads are usually output from sequencing facilities as text files in a format called FASTQ. Okay? Now, this format depends on an earlier format called FASTA. Now, the FASTA format was developed as a text-based format to represent nucleotide or protein sequences. Now, the FASTQ format is designed to handle base quality metrics output from sequencing facilities. Now, once you have your raw reads, you'll now proceed with computational pre-processing, which involves quality control and trimming. Now, the raw reads will then be subjected to quality control and trimming, and during this process, sequencing artifacts such as low-quality reads, contaminating reads, and as well as adapter sequences are removed. So once you do that, you will now have your clean reads. Okay? So raw reads, and then quality control and trimming, and then you have your clean reads. After that, in order to identify genes in these clean sequences, these sequences have to be compared against known genes. Okay? And this is done by a combination of assembly-based and reads-based approach. Now, the reads-based approach or method align or map clean reads to curated databases and output feature tables, whereas assembly-based methods assemble clean reads into contigs before performing taxonomic and functional annotation and mapping. So here, we have, after that, after you perform analysis, ano yung gusto mong malaman? What are the two questions that you want answered after doing all this analysis? So we have two questions that we want answered. Okay? This is the example of the raw read, by the way. And the two questions that we have to answer, or that we will be able to answer after this analysis, is that who is there? Sino ba yung nandun sa sample mo? Sino yung nandun sa environment na yun na inaaral mo? Okay? So who is there, and what are those organisms doing? So what are they doing? So ito yung dalawa nating makukuha from metagenomic analysis. So who is there and what are they doing. Basically, this is taxonomic and functional profiling. Now, it is hypothesized that karst ecosystems support abundant and diverse microbial communities that include ecologically, agriculturally, and economically relevant taxa, and the baseline data generated from this study may provide valuable insight on the karst ecosystem and may pave the way in long-term monitoring of microbial diversity. The objectives of this study, of this project, is to describe the taxonomic and functional diversity profile of bacterial and fungal communities of karst soil samples using NGS and metagenomic analysis, and to relate the distribution and function of bacterial and fungal communities to karst ecosystem health and conservation. All right. So just to give you an overview of the methodology, so first you do soil sampling. After soil sampling, perform DNA extraction, then sequencing, NGS, and then metagenomic analysis. So this is the sampling area. This is the Samar Island. And we actually have 4 sampling sites. But for this talk, I'll just be focusing on one specific municipality, and that is Basay. So ito lang po yung ipepresent kong result for this specific talk. So as shown here, you can see the geographical location of sampling in the karst forest. It's labeled B1, B2, and B3. So this is in Basay, Samar. So what we did was we first collected the samples, and after collecting the soil samples, we then sent them to the laboratory for immediate processing. So DNA extraction was performed using a soil DNA isolation kit following the manufacturer's protocol, of course. And then the DNA samples were then checked using gel electrophoresis. So the quality of the DNA, of course, was checked with or using gel electrophoresis. And then the DNA samples were then submitted for next generation shotgun metagenomic sequencing. This metagenomic shotgun sequencing was performed by First Base Laboratories in Selangor, Malaysia, using an Illumina MiSeq instrument. So the platform, the sequencing platform that we used was Illumina MiSeq instrument. Now, after obtaining the raw reads from Illumina MiSeq, so these raw reads were then processed. So kung alalahanin nyo kanina yung diniscuss natin na is a simple diagram on a metagenomic workflow. So once you have your raw sequences in FASTQ format, what we did was we uploaded the data. So data was uploaded as FASTQ files, and then the paired end reads were joined. And what we used was the metagenomic rapid annotations using subsystems technology, or known as MGRAST. Okay? So ito po yung ginamit namin na metagenomic pipeline for this specific study. Now, after uploading the FASTQ files and the paired ends, joining the paired ends, the joined paired ends were then subjected to this pipeline. Now, the uploaded data were then pre-processed, or computational pre-processing was done using Solexa QA. So ito yung na-mention ko kanina na we do trimming and quality control. So what we used was Solexa QA in order to trim the low quality regions from the FASTQ data. Right? And then after which, the replication was then performed using a simple K-mer approach in order to remove artificial duplicate reads. Now, these ADRs were then analyzed using duplicate read inferred sequencing error estimation. I know it's a mouthful, so it's DRI-C. And this is just done to determine the degree of variation among prefix identical sequences in the template. After which, the sequences were then screened using bowtie, and gene calling or gene prediction was performed using FragGeneScan. And this is used to predict the proteins or protein fragments from de novo sequence data. Then in order to preserve the relative abundances, Chi-Me was used to build clusters of proteins at the 90% identity level. Then a representative from each cluster, or for each cluster, was then subjected to similarity analysis. And then sequence similarity searches were then computed against a protein database which is derived from the ND5-based non-redundant protein database, or M5NR. So the RefSeq database from NCBI was used for taxonomic assignment, while COG, or cluster of orthologous genes, then KO, which is KEGG orthologs, nog, and seed subsystems were then used for functional assignment. So for our results, across all samples, after the analysis of domain bacteria, a total of 28 phyla, 51 classes, 110 orders, 244 families, and 596 genera were detected. As for the fungal groups, a total of 6 phyla, 18 classes, 42 orders, 92 families, and 132 genera were detected. The most dominant taxa that we detected were actinobacteria and proteobacteria. So in all karst soil samples, B1, B2, and B3, there is an abundance of firmicutes as well, acidobacteria, chloroflexi, planktomycetes, cyanobacteria, veromicrobia, and bacteroidetes. Now, all other groups that were not mentioned are of the minority. So again, the most dominant taxa that we observed or that we detected were actinobacteria and proteobacteria. Now, proteobacteria, being a group of microbes that are capable of responding to unstable carbon sources, they were also found in other karst ecosystems. Now, this is supported by one study in China made by Yuan and his colleagues in 2016, where they reported that at the phylum level, they also detected actinobacteria and proteobacteria as dominant phyla in that specific karst ecosystem. Now, within the proteobacteria group, alpha proteobacteria were the most abundant. And among the alpha proteobacteria, various genera including Bradyrhizobium, Rhizobium, and Nitrobacter, which are responsible for nitrogen cycling, were found to be abundant across all sampling sites. Now, the relationship between legumes and Rhizobium as well as Bradyrhizobium bacteria are considered by far the most important nitrogen fixing symbiotic associations. On the other hand, Nitrobacter, which is one of the most studied nitrite oxidizer under the alpha proteobacteria group, utilizes the nitrite oxidoreductase, which is an enzyme that we actually detected pa din dito sa study using KEGG orthologs or KO. So this is used for nitrite oxidation. Now, this goes to show that there are microorganisms that could possibly have plant growth promoting abilities or plant growth promoting traits. Now, just to show you our analysis of the sequences, so analysis using KO reveals the presence of functional proteins and enzymes that are involved in nitrogen cycling. As you can see here, there are many different colors that you would see. So you could see 3 colors that are assigned to each specific site for B1, B2, and then B3. So you can see here enzymes which are present in all sites that are involved in nitrogen metabolism. So you can see here colored EC numbers which are identified in the 3 karst areas in Basay, Samar. Now, what are the other enzymes that we detected? So first, we detected also, actually marami po kami na-detect. I just chose some of the important enzymes that I think are essential. So first, we have 1-aminocyclopropane, 1-carboxylate or 1-carboxylic acid deaminase. Now, bacteria with ACC deaminase activity has the potential to promote plant growth development and growth under adverse environmental conditions. Now, the increment in synthesis of ethylene from its immediate precursor, which is ACC or the 1-aminocyclopropane 1-carboxylate, secreted by plants as root exudates, has been found in almost all plants growing under stress conditions. So mataas po yung ethylene dun sa mga plants that are growing under stressful conditions. Now, ano po yung importance nito? ACC kasi, sinabi ko kanina, is its immediate precursor. Right? Now, ethylene, we know, most of us know probably, is a plant hormone that is involved in regulation of various physiological processes of plants. But stress-induced ethylene, okay, in plants inflicts a significant reduction in plant growth and development and can lead to plant death. Now, it is widely reported that certain bacteria that possess ACC deaminase enzyme can degrade ACC, which is the precursor of ethylene, to ammonia and alpha-ketobutyrate, thereby reducing the level of stress ethylene inside the plant. So that's one of the enzymes. Then another enzyme is a phytohormone or enzymes for biosynthesis of auxin or IAA. So enzymes involved in IAA biosynthesis were also detected, such as indole 3 acetaldehyde oxidase and indole pyruvate decarboxylase, which is this auxin or IAA is actually a signal molecule that is involved in the regulation of plant development, such as organogenesis, trophic responses, cellular responses such as expansion, division, and differentiation, and other gene regulation. Now, this phytohormone is synthesized from L-tryptophan, and different pathways are involved in this synthesis. Now, we also detected acid and alkaline phosphatases, which are able to release free orthophosphate from recalcitrant organic phosphorus forms, thereby improving the bioaccessibility of phosphorus. Then several genera of interest under the gamma proteobacteria group were found to be abundant in the sampling sites, and this include Escherichia, Haemophilus, Salmonella, and Klebsiella. Now, these bacteria were found to produce the enzyme carbonic anhydrase, and again, this enzyme was also detected during the functional profiling, which this carbonic anhydrase is a metalloenzyme that catalyzes the hydration of carbon dioxide to bicarbonate and protons. Now, the said enzyme participates in two chemical reactions such as corrosion and precipitation, and these actually are considered as key processes in the karstification process. The most dominant genera in actinomycetes or in actinobacteria was detected to be Streptomyces. So I know we're all familiar with Streptomyces, aside from it being known as the most significant source of biologically active compounds. Streptomyces are also ammonifying bacteria, and they can be found in soils, and they aid in the decomposition of organic materials. They were also found to be capable of inducing calcification through at least one biomineralization pathway. Now, molecular studies indicated as well that the genome of acidobacteria have genes involved in the carbon, nitrogen, and sulfur cycles, as well as those required for the degradation of complex polysaccharides. Now, the group of autotrophic green non-sulfur bacteria responsible for carbon dioxide fixation, which is the chloroflexi, was also detected. And some other autotrophic bacteria, nitrospirae and chloroflexi, might be also involved in the transformation of the nitrogen element into the nitrogen cycle by the nitrogenase NIFH enzyme and utilize inorganic compounds by nitrification. Then we also have planktomycetes, which play a role in carbon and nitrogen cycles. Now, based on the RefSec database, the number of sequences affiliated with each fungal taxon in the karst soil were similar across all samples. And the dominant phyla in the karst soil metagenome is Ascomycota and Basidiomycota. And then the most dominant fungal classes are those of Soridariomycetes and Urosomycetes, right? So in all areas, there is also abundance of other groups such as Saccharomycetes, Agaricomycetes, Dothideomycetes, and others, right? So in karst ecosystems, ano yung role naman na piniplay ng fungi? So fungi play a role in many important environmental processes such as organic matter decomposition, protection against leaching by element storage in biomass, and element released by mineralization. So in one study actually by Lian back in 2010, the role of fungal mycelia on accelerating the breakdown of carbonate rock was described. Fungal mycelia inserted into crevices and absorbed moisture from the microcracks in the rock, which led to full dissolution of acid secretions of bacteria within the rock. And then in addition, some fungal strains can utilize calcium nitrate as nitrogen source via the reduction of nitrate to ammonium by nitrate reductase and nitrite reductase as well. So these two enzymes are actually detected as well when we did functional profiling. This leads to an increase in the pH. So relatively neutral yung pH na na-observe namin actually dun sa ating mga soil samples from karst. Then the calcium ions in the medium react with carbon dioxide released by fungal respiration or with bicarbonate in order to form calcium carbonate. Now, based on KO, ito yung heat map ay showing the functional profiles of the microbial metagenomes of the soil from the karst soil in Basay Samar, and we saw that metabolism was the most abundant. Now, earlier I showed you the enzymes involved in nitrogen cycles, in the nitrogen cycle, and that's just one of the many pathways actually that were detected upon studying the metagenome. So other pathways include the reductive carboxylate cycle and the sulfur cycle. So as you can see here, enzymes that are involved in the reductive carboxylate cycle in photosynthetic bacteria were found in all areas in Basay Samar. So makikita nyo dito, all of the areas would contain these enzymes that are important for the reductive carboxylate cycle. Same goes with sulfur metabolism. So we found enzymes that are essential for the sulfur metabolic pathway in all three sites. Now, to summarize, the most dominant bacterial taxa again were actinobacteria and proteobacteria, and the most dominant fungal phyla is Ascomycota. Then the most dominant fungal classes are Sordaromycetes and Urosomycetes. Now, the karst forest in Basay Samar supports a diverse ecosystem as evidenced by the presence of various bacterial and fungal phyla which perform specialized functions in nutrient cycling. And this pioneering study on taxonomic and functional diversity of microbial communities or bacterial and fungal communities was made possible using next generation sequencing using the Illumina MySeq platform. So the results highlight that a significant link between phylogeny and functional potential. Analysis of the metagenome reveals potential metabolic pathways for the metabolism and cycling of carbon, nitrogen, and sulfur. So for our recommendations, future research may focus on elucidating the interaction of these microorganisms with other organisms such as plants in the karst area. So what we could do, if you would remember yung previous talks on plants, animals, so we could relate the microorganisms dun sa mga plants present in the karst ecosystem, and then studies on how they could affect the survival of the species in the ecosystem may be done. And as the current study provides a baseline for the karst metagenome, comparison with other ecosystems may be worth exploring. So the project would like to thank University of the Philippines Los Baños, UP Manila, Samar State University, DOST-PCAARRD, SINP, then the local government units of Paranas, Taft, Basay, and Giwan for allowing us to study there, and then Basaranan nga Organisasyon Han San Isidro Samar or BOSIS. That's all, and thank you for listening.
Thank you, Prof. Daniel, for sharing with us your webinar on the bacterial and fungal communities of Kaigangan Forest. And as an act of appreciation, we will give you the certificate, which will be emailed to you. Okay. And let's now proceed to the question and answer portion today. And we will helping us in this part of this program is Prof. Des Fernandez. And again, I'd like to remind you that if you have any questions, you can leave them in our chat box or in the comment sections for those who are in Facebook Live. And I'm also quite glad to see 85 participants here in Zoom. So imagine we had such a high turnout today. And also, I'd like to begin sharing with you the link for our evaluation. So here is the link for our evaluation form. We have bit.ly/kaigangan week 5. So please remember that bit.ly/kaigangan week 5. And please note that this link can only be accessed until 1 p.m. today. Okay? So yeah, just let it stay here for a few more seconds before we proceed to our Q&A. Okay, you get the link. We'll also put it in the chat box and the comment section. Okay, and now I'll pass the spotlight to Ma'am Des to go with the Q&A.
Thank you very much, Elaine. Rinig ba ako? Okay naman.
Okay po.
Okay. And thank you very much, Sir Daniel, for your enlightening talk. So we will now proceed to answer some questions from our audience, and we'll accumulate the questions sent in advance first through the registration form, and then those from our Zoom and Facebook Live comments. Okay. Hi, Sir Daniel.
Hi.
We have a question from Damsel Bangkal from IPB UPLB, and the question is, what next generation sequence platform are you using? I think you already mentioned this, but maybe for the benefit of our students, you can explain more about this method.
So as I've mentioned, the sequencing platform that we use is the Illumina MySeq. So may iba't iba kasing sequencing platforms. So may Illumina MySeq, may HiSeq. So different sequencing platforms can be used. And what we used was Illumina MySeq, which produced paired end reads. So I think medyo isang mas mahabang lecture pa kasi if we discuss more about the sequencing. But basically, like what I mentioned, the difference between the NGS and the Sanger would be sequencing of a large number of samples, a large number of targets at one single time. So maaaral mo agad in just one sequencing experiment, yung lahat ng yun, yung mga present ko kanina, lahat ng mga actinobacteria, etc., lahat ng phyla na yun will be detected in just one run.
Yeah. So it's very efficient in that case.
Yes.
Okay, so thank you very much, sir. And our next question from Maria Elenita de Castro from UPLB. What are the direct impacts of the identified soil microorganisms on the growth of the tree species in the limestone forest?
All right. So in the presentation, I mentioned some of the possible impacts of SMCs or soil microbial communities in the karst ecosystem. And we found out that some of the microorganisms that could potentially be involved in nutrient cycling, such as some genera from the proteobacteria phyla, and there are those which could be involved in the production of antimicrobial substances such as your streptomyces. And there are those, so kung may streptomyces ka, of course, it could possibly have bioactive compounds that could inhibit some phytopathogens. And another would be the plant growth promoting na mga enzymes, so ACC deaminase, like what I mentioned, enzymes for producing phytohormones and the like. Now, as to the direct impact of a specific genera to a specific tree species in the forest, that could be another project, I think, or study in the future. The rhizosphere of certain plants or tree species can be studied in more detail. So if that's ayun, kung yun po yung specific species ng bacteria or organism sa specific tree species. Ayun po.
I think siguro kasi a lot, apart from yung syempre na-mention natin yung benefits, siguro most people are thinking of microorganisms as bad, lalo na ngayon. Pero they actually have a lot of benefits for our ecosystem. And the next question is from John Paul Ramos from DLS-MHSI. How does the profiling of soil microbes around the root system of plants endemic to certain areas give insight on how to construct or maintain or optimize a sustainable setting for crops common in the Philippines, such as rice and corn?
All right. So now that we have the baseline data for the soil bacteria and fungi that are possibly present in this type of environment, and we also were able to elucidate the possible roles of these organisms in this type of environment or ecosystem, we can actually try to isolate some of these microorganisms and screen for their plant growth promoting abilities, such as nitrogen fixation, ACC deaminase activity, production of phytohormones, and phosphate solubilization. And then after the screening process, we can get the top isolates and we can actually make biofertilizers or fertilizers that could benefit these common crops such as your rice and corn.
Mm-mm. Yeah, and I think, and may follow-up question din si John Paul na, what can you say about yung potential of these soil microbes for drug discovery for pathogens in animals and also humans?
Ayan. Actually, this is a very interesting question na um, this in this study, we were able to detect high numbers of actinobacteria and many species of which would be capable of producing these bioactive compounds. And it's possible that there are some novel compounds na hindi pa na-discover since this is not a well-studied ecosystem, this karst forest. So it's possible na merong mga bioactive compounds that are novel which could be extracted from the microorganisms or species in this type of environment. But anong isa, when it comes to identification of potential pathogens of humans, plants, and others. Now, very interesting, two of our UP Manila students actually are studying the resistome. So in the resistome, it includes yung antimicrobial resistance genes, which could be present in some of the pathogens that are present in that specific environment. Now, if you would want to know more about the possible or the resistome, specifically of the karst ecosystem, then pwedeng hintayin natin yung publication na ating mga students.
Mm-mm. Wow. And speaking of learning more, our next question is from John Raymond Torres from DMMSU, and I think either a prof or maybe student siya. And he's asking, can you please suggest a small-scale research that our students and novices in this field can do or follow so that they can contribute to a collaborative work maybe?
Right. So, well, we in Project 3 suggest that you can start with being trained on how to analyze this microbiome. Okay, so this microbiome data like what we have now. Now, there are actually many research questions that can still be generated from this study. And this might be small, itong pag-training, but this is also collaborative and yet has a strong impact to science and society. Okay? And actually, this mid-year, I talked with Dr. Marilyn Balolong from UP Manila. I was told that UPM will conduct a training on metagenomic analysis and understanding these pipelines. So what you could do is you could join the practicum students of AMHERG UP Manila. You may contact Dr. Marilyn Balolong. I could give you her contact details. And yeah, so I think that's it.
Mm-mm. So yun, meron palang practicum students if you're interested. And also, if you look at yung background, virtual background ni Sir Daniel, you can see there the email address of CONserve-KAIGANGAN, and you can contact the program and we will direct you to Project 3 for that. Our next question is from Lindsay Susie, and her question is, what happens when there is an overlap between the niches of fungi and bacteria as it relates to ecosystem health or functioning? So that's very interesting.
Hey, overlap between niches of the fungi and bacteria. All right. Now, I think generally speaking, for all environments, bacteria, fungi, and even archaea or even protozoa, they would be paano ba, parang mayroong check and balance sa kanila na sa pagkakaintindi ko dun sa question is as it relates to ecosystem health or functioning. It's possible naman na nagtutulungan sila, I think, in a way, or some might be neutral lang. For example, like what I mentioned, actinobacteria, such as streptomyces, are capable of producing antimicrobial compounds which could inhibit the phytopathogens present in the environment. So that's one overlapping instance. Okay, so makikita natin pwedeng affect yung ating phytopathogens. Another one would be, I think, yung sa nitrogen cycling. So when you have nutrient cycling, there are some microorganisms, for example, in the nitrogen cycle. So there are those which are involved in N fixation. Tapos yung products naman nun can be used by another organism and so on and so forth. So that goes for all the other nutrient cycling, I think.
Mm-mm. And I think kasi limestone forests are so understudied that there's a lot more to learn, particularly in this topic na yung interactions between bacteria and fungi siguro.
Exactly,
So
yes.
maybe there are interested students out there who want to explore that.
Yes, it's actually very, napakalaki kasi talaga ng data. The data is actually overwhelming that you could really get so much data from it and maraming pwedeng aralin pa. So, hindi lang basta, it doesn't stop here, itong mga na-present ko sa inyo. Like what I said, some of the UP Manila students are studying the resistome. We can actually proceed with identifying specific antimicrobial resistance genes, resistance to heavy metals, etc. So marami pang pwedeng aralin.
Yeah. And since ngayon na hindi makakapag-field yung mga students natin, for example, sa amin sa Project 1, we also have a lot of data that we are now opening to students if they are interested na baka gusto nyo i-analyze tong part na to, itong part na to. So the whole program is opening to that kind of collaboration na from the comfort of your own home, you can do your research. And we have another question here from our very own Sir Giro Adorador. So indeed, this research is among the first of its kind in the country. What would you consider to be the most surprising discovery of this study?
Most surprising discovery of the study? Well, to be honest, I was already expecting some like plant growth promoting traits because it's very, well, you could say that it's a stressful environment for plants. But I'm expecting a relationship between plants and the microorganisms, bacteria and fungi alike. But siguro yung surprising would be baka matakot sila, no? Hindi ko alam kung dapat ko sabihin. But like for example, we were able to detect Mycobacterium na genus. So we know that Mycobacterium genus, specific species of it would be Mycobacterium tuberculosis, which is the causative agent for TB. So we detected Mycobacterium, but of course, Mycobacterium also has some positive impacts on the ecosystem, such as in the cycling of nutrients as well. So that's why na medyo siguro kung surprising, nagulat. Kasi in terms of the beneficial microbes, medyo kind of expected na meron nun. But siguro another one would be microorganisms involved in the karstification process. So like what I mentioned, some members of the gamma proteobacteria such as E. coli, Haemophilus, etc. Would be capable of performing chemical processes that could be involved in the karstification process.
I think, well, in also my own research with diseases sa wildlife, syempre marami tayong mga zoonotic diseases, and you can also get, pathogens from the environment. So I guess maybe that also justifies not parang destroying the ecosystem of yung karst environment. Kasi syempre we don't know what we will uncover and how it will affect the whole ecosystem. So yun. And we have a question about filtering noise. So from Gardel Saiza Labunao, how did you filter noise or the contaminating DNA outside your sample of interest?
Now, as I mentioned, we used a relatively sobrang tawag dito, user-friendly na metagenomic pipeline. It's MGRAST. You can actually do it at the comfort of your own home. You don't need a supercomputer to use MGRAST. Ako actually some of it ginamit ko lang using my laptop. Kasi sa MGRAST kasi dun sa kanilang site, you upload it and ang ginagamit mo actually ay yung system nila. So ginagamit mo yung system nila. And then, in that specific pipeline, nandun na yung, kasama na dito yung filtering ng noise or contaminating na sequences, na hindi mo gusto. So, andito nga yung sinasabi natin kanina na the computational pre-processing wherein you filter the noises using, specifically for MGRAS, it's Solexa QA. Yun yung ginamit. But you can also use, tawag dito, Fast QC, pwede nyo yung gumamit. So, pwede nyo gamitin. Maraming ibang mga methods. But again, the important thing is you research first what are the pipelines being used in your specific site or area of study. For example, kung soil, sediment, aquatic, so or gut microbiota, for example. So ano yung usual ginagamit ng mga scientists? And you can pull them in or even use again or copy the pipeline mismo na ginamit nila.
Yeah, I think yan yung mga one of the biggest worries ng students who are just starting out in mall bio, yung about contamination. So research your pipelines. That is the key. Okay, we have a question from Mark Angelo del Rosario. Good day. Thank you for this insightful experience. Can this method of analysis also be applied to novel substrates of microbial communities like platyspheres in marine water? So hindi ako familiar din sa term. So baka you can say something about that.
All right. To be honest, I'm not also familiar with platyspheres, but I would just be using context clues for this. How can this method of analysis be applied to novel substrate of microbial communities in marine waters? So what I would be able to say is that kahit anong environment, whether it's soil, whether it's marine waters, fresh water, you can do metagenomic analysis. So you can do taxonomic and functional profiling. It's not limited to terrestrial habitats. So meron siya pwedeng sa gut, sa animals, sa plants. You can apply this in all different living things. Di ba?
Yeah. So there's a lot more out there to discover. And our final question is, I think I can also answer this from Rick Quinones from LGU Hernani. Hi, sir. Have you conducted biodiversity studies specifically here in the municipality of Hernani? So right now, I think na-mention naman ni Sir Daniel na we only have 4 na naging 3 sites. And then after the pandemic, medyo natigil kami ng fieldwork. But if we have phase 2 of the project, of course, we would love to explore more of Samar kasi ang laki nga po nung forest over limestone in the island. So we would love to do that. We have pahabol pa na question. I think this might be our last question. From Gabriel Joshua Tyson. Hello, I'm Gab from CFNR UPLB. I would just like to ask, when will the paper be published? I'm currently studying a related subject regarding the bacteria and AMF. What do you think is the best soil sampling method for studying soil microflora? So I think maybe our students are also interested na parang, how long does it take to publish a study like this and how difficult is it? Parang ganun.
All right. Kailan? I can't really say kung kailan. We sinend na namin for publication. We already sent 1 paper for publication. We're working on, actually 2 na pala. May sinend kaming 2 for publication. Kailan ina-expect? It depends really on the journal na pinagsubmitan mo.
Yes.
Kapag, for example, kasi may mga journal na mabilis mag-reply, merong mga medyo mabagal. It also depends on the impact factor of the journal. Kung, for example, kung yung study mo ba merits a slot, kumbaga, sa high impact factor journal or something like that. But, ayun, dadaan pa kasi siya sa review, etc. So, I can't really say when. Hopefully this year, hoping. But naipasa na namin po 2. And kung interested naman po kayo dun sa resistome, isisulat pa po yan ng aming mga babies from UP Manila. Ayan, so ayun pa po. Tapos meron pa po kaming, yung 2 naming master student na meron po silang, inaaral din po nila yung sa Paranas naman po and dun sa Taft na area.
And I think yung second question niya is, what is the best soil sampling method for studying soil microflora? Although I think na-mention na siya.
Well, best soil sampling method, well, it actually depends. So merong proper way of soil sampling. But in the karst forest kasi, hindi malalim yung soil. So kung maaalala natin, tayong mga pumunta doon, di ba, na mababaw lang po yung soil. Hindi mo siya kayang hukayin ng several centimeters. Sobrang manipis lang po. So what you could do depending on the soil sample, um, depending on what your site is kasi eh. If you want, you could contact me. We could perhaps exchange some emails regarding your concern. But it varies eh. Pero merong mga standardized na soil sampling. But again, you should adapt to a certain environment. Say for example, sa karst, manipis kasi yung soil.
Yeah.
Ayan.
Yeah, so if you want to contact Sir Daniel, sir, are you willing to give out your official
Yes,
address?
my email is, I'll just type it here.
Okay, so while Sir Daniel is typing his email address in the chat, maybe we can entertain one more question, our very last question. Last na to talaga. So, good day, sir, from Bonnie Datul. Good day, Sir Daniel. Congratulations on your results. What variable region of the 16S RNA gene did you target for the metagenome sequencing? How did you incorporate the biases with the said region in your data analysis? Thank you.
All right. Variable regions in 16S RNA gene. To be honest, I can't remember dun sa sequencing result na sinend sa amin ng first base. I can message you after this, Bonnie, with regards to what variable regions. I'm not sure if it's V3, V4 regions of the 16S. Then how did you incorporate the biases with the said region in your data analysis? Again, we just followed yung MGRAS na pipeline, which is marami actually publications na using this this metagenomic pipeline. And I think it's very user-friendly. The students, I think even BS students can use this. Actually, ginamit nga ng UP Manila students yung MGRAS as well. And you can use other platforms such as Metaflan, etc. Marami pa po.
Okay, thank you very much, Sir Daniel. And thank you very much to all our audience members who asked their questions. You can contact Sir Daniel with email address jpong2@up.edu.ph. Huwag niyo po i-contact si JPong or si JPong1.
Hindi ko po kilala yun.
Hindi po sila si Sir Daniel. Okay, and I will give the floor back to Elaine for the closing of our program.
So thank you, Prof. Daniel Ong, for answering our questions for our audience. And we'd also like to thank Ma'am Des Fernandez for facilitating the Q&A portion on today's webinar. And most of all, we would like to thank our viewers in Zoom, Facebook Live, and YouTube in the future for spending some time to listen and learn with us. And again, we would like to reiterate and request everyone to fill out our evaluation form. That's bit.ly/kaigangan week 5. Okay, again, I'm showing it again. So for those who haven't filled out our form, so here's the link. You can scan the QR code and it's only accessible until 1 p.m. only. All right. So indeed, we had a very fruitful discussion this past few weeks. So we have started at around mid-May to our webinar series, and we are now on our week 5. And we have seen and learned a lot about biodiversity of Samar and its value, for which leads us to conclude that we really need to conserve and use our natural resources sustainably. And before we end this program, let us all hear from Assistant Prof. Marjorie de Los Angeles of UPLB as she provides a synthesis of our webinar series.
So, hello everyone. Thank you very much, Elaine. Allow me to share my screen. Okay, so this is relatively a short presentation just to synthesize what has been happening for the past few weeks. So as we conclude the webinar series for CONserve-KAIGANGAN, on behalf of the program, thank you again, everyone, for joining us. So just a quick recap, what were the different topics that were tackled from week 1 to today, week 5. So here are the different topics. So week 1, we had potentials of economically important native plants of Samar Island, Philippines, delivered by Assistant Professor Marlon P. Rivera. And a second talk on the same week entitled Biodiversity Research for National Development delivered by our very own program leader, Dr. Inocencio Ibo Jr. Week 2 was a topic regarding biodiversity of forests over limestone in Calicoan Island, Giwan, Eastern Samar by our very own project staff, Assistant Professor Desamarie Antoinette Fernandez. And for week 3, we had the topic on mother trees of Samar Island, karst forest, identification and community-based conservation by Miss Jessa B. Madera. Week 4, last week, we had biodiversity of forests over limestone ecosystems in Samar Island Natural Park, delivered by Miss Elaine Lorene Villanueva, alongside with me, where I delivered the topic Ecology of Understory Plants of Forests over Limestone in Samar Island, Philippines. And for today, we listen to the talk delivered by Assistant Professor John Daniel P. Ong entitled First Report on the Profiling of Soil Bacterial and Fungal Communities in Karst Forests of Samar using Next Generation Sequencing. Such a novel research. Now let's look at how many of you stuck with us till the end. So looking at the statistics of the webinar per week, week 1, we had 44 Zoom participants with peak viewers at 16 and 53 evaluation responses. Week 2, we have 60 with 19 FB peak viewers and 91 evaluation form responses. Week 3, we have 49 Zoom participants, 36 FB peak viewers, 145 evaluation form responses. Last week, we had 67 participants, 23 FB peak viewers, and 89 evaluation form responses. For today, I think, Sir Daniel Ong, nasa iyo ang corona. Mukhang we have 85 Zoom participants. I don't know how many FB peak viewers we have, and evaluation form responses. Now, again, where to next? So as we wrap the webinar series and we are all introduced to the different outputs of the varying projects of the program, what are the different, where to next, or where is the program leading to? So in cognizant to the UN Sustainable Development Goals, the program has yet to achieve a number of sustainable goals, such as number 1, no poverty, 0 hunger, quality education, decent work and economic growth, sustainable cities and communities, responsible consumption and production, climate action, life below water, life on land, and partnership for the goals. So far, the project has been able to document a number of flora and fauna, understory species, our mother trees, soil microflora, and so on and so forth. Now, aside from the various scholastic outputs, here are some of a few, if you haven't read them yet, from the different study sites that we have. So first, we have the one scholastic output as a product of Project 1. So biodiversity in forests over limestone in Paraná Samar Island Natural Park, a UNESCO World Natural Heritage Site nominee, authored by members of Project 1 and led by Ms. Elaine Lorene C. Villanueva. So here in the slide showing the notable species of plants and animals. So some of which are new locality records for Paranas for the plants, Plactaria calcarea, Artocarpus rubrovinus, and Hansea wenzeliana. So for Paranas, a new locality record for animals is the Platymatis bayani. Aside from this publication or scholastic output, we have another one, Flora and Fauna Inventory of Limestone Forests in Taft, Eastern Samar, Philippines, authored by Project 1 members and led by Ren Vivian R. Obenya. So here are some, again, photographs of notable plants and animals. If you are interested to read these publications, these are all open access and are available online. Third, we have fauna and flora of forests over limestone in Calicoan Island, Giwan Marine Reserve Protected Landscape and Seascape, Eastern Samar, Philippines. Again, written by members of the Project 1 and headed by Miss Desamarie Antoinette P. Fernandez. Aside from the various scholastic outputs, we have also produced a lot of communication education materials in the form of SEPA. So here are some of them. Again, they are readily downloadable. If you access our, or if you send us a message, we are happy to provide you with copies. Aside from SEPA materials, the program has also developed an undated planners. This 190 pages of colorful with colorful photographs of species that are found in the area. Per month, there we highlighted notable flora and fauna that are found in Samar Island. So aside from these different scholastic and SEPA materials, the project is not yet done. So the program is not yet done, essentially, as mentioned by Sir Daniel Ong and Ma'am Des Fernandez. So there is still a lot of ground to cover, a lot of forest floor to cover. So we are encouraging everyone to collaborate with us. As mentioned earlier, there are a lot of data sets already ready to be analyzed. So the program is open to your help, your collaboration to help us understand further the forests over limestones that are found in Samar. So aside from the unique fauna, there is also a number of unique plant species that we have yet um, to describe, to discover, to record, to document, and with your help, with the program, then we will be able to attain the development goals that the program is trying to achieve. So, and with this, you can communicate with us, or you can shoot us an email using the following. So you can visit our website, kaigangan.uplb.edu.ph, or follow us at fb.com/concaigangan, or send us an email, kaigangan.uplb@up.edu.ph. So in behalf of the members of the program, as seen here in this photograph, thank you everyone for lending us your ears, lending us your time. Thank you to all of our LGU partners, and our People's Organization partners, our collaborators, friends from the museum, Kuya Danny, and all other of our colleagues. So once again, thank you very much, and that is all. So Aling, thank you once again. You can now have the screen.
Okay. So thank you, Mom Marge, for wrapping up our what happened during the past 5 weeks. And we would like to thank everyone, our participants for today, for being here and for being with us, and for those who have stayed this past 5 weeks and watch with us. Thank you very much. And for those who will watch in the future of this webinar series, we would also like to thank you for spending some time to learn about the forest over limestone of the Philippines. And again, I would like to share with you for the last time the link for our evaluation form. So that's bit.ly/kaigangan week 5. Okay, and this is the QR code. You can scan it also in case you have this QR scanner in your phone. And this can only be accessed until 1 p.m. only. And also for our webinar series, in case you missed those, so remember, we have here the link for our YouTube playlist. So we have bit.ly/kaigangan playlist. Okay. And that's all. And before we end, we would like to request everyone to show your screen, and let's have a photo for one last time. Okay. Okay, hello everyone. Ayan. I'd like to see your beautiful faces. Okay, so we have here, how many pages do we have here? We have 3 pages. So while waiting for the others, again, I'd like to remind you of the evaluation form. That's bit.ly/kaigangan week 5. Okay. So let's now take a photo. So we have 3 pages here. So let me show your smiles here. Okay, first page. Okay, smile. So we have 1, 2, 3, smile. Yeah. Okay, I'm now proceeding to the next page. Okay, keep smiling. Okay, 1, 2, 3, smile. And lastly, we have your page 3. Okay, 1, 2, 3, say smile. All right. That's all, and thank you everyone. And in case you would like to be with to stay in touch with us, we have here our um, our link for our here. I'd like to show you again our link so you can reach us via email, via Facebook, and our YouTube channel. Okay, and that's all. Thank you everyone for watching our webinar series, and we hope to see you in our future activities. Okay, bye. Bye everyone.